Good morning everyone, my name is Janatul Jafre. Today I'll be presenting our study on expression of tropomyosin 2 TPM2 in HK29 cell using pcDNA 3.1 plus vector. This project focuses on understanding how TPM2 contributes to cytoskeleton integrity through action filament stabilization. Purpose of the study. The primary goal of the study was to clone and express the TPM2 gene in HK29 cell with the pcDNA 3.1 plus vector. We used pcDNA 3.1 plus vector driven by CMV promoter to examine the role of TPM2 in action filament stability and cytoskeleton organization. The result aims to contribute our understanding of cellular morphology and mutability mechanism. For this slide, this figure shows an overview of cloning and expression workflow. The TPM2 vector was first inserted into pcDNA 3+1 vector, then introduced into HK29 cell for expression. This setup allows the TPM2 protein to be expressed under the control of CMV promoter, which ensures strong transcription in mammalian cell. Now E. coli transformation for pcDNA 3.1 plus TPM2 plasmid amplification. Before introducing the construct into HK29 cell, we needed to amplify the TPM2 containing vector in E. coli. Component cell were transferred via heat shock, then recovered into LV medium and plated on ampicillin containing agar. Colonies were screened to identify successful transformant, which were then used for plasmid extraction. Now the verification of accurate vector formation. After amplification, it was essential to verify the accurate formation of the recombinant plasmid. So this could involve methods such as restrictionization, PCR or sequencing to ensure that TPM2 gene was inserted correctly and in frame with any design tags. Now TPM2 gene and the protein expression comprehensive verification. For this, once transfected into HK29 cell, verification of TPM2 expression was crucial. This could be done through qPCR for TPM2 mRNA, Western blotting using anti-TPM2 antibodies for protein detection, or fluorescence microscopy if the protein was tagged with flag or GFP. A successful expression confirms that the CMV promoters is functioning and that the HK29 cells are producing the target protein. Here is the reference that supported this study, including previous research on TPM2's role in cytoskeleton dynamics and even its implication in cancer. These works provided both a foundation and validation for our experimental design and interpretation. Now the conclusion is, we successfully cloned and expressed the TPM2 gene in HK29 cells, enabling us to study its role in action, filaments, stabilization, cellular structure. So this work may pave the way for deeper insights into cytoskeleton disorder and potentiality in cancer biology. Thank you so much everyone.